Welcome back, I'm that Chemist, and today we're going to be deciding which chemical is the worst neurotoxin. Before we can discuss the neurotoxins in this video, first we need to discuss what nerves are and how they fundamentally work. Now, if the discussion of nerves makes you a bit nervous, rest assured that this is interesting, even if you aren't feeling very stimulated. In fact, your lack of interest couldn't be more relevant to this video. See? Now you're listening. Why did I pause? Why the sudden change in audio signal? One way that we send signals throughout our body is via our nervous system. Our nervous system is comprised of approximately 86 billion neurons, and you can't just round it up to 100 billion neurons because 14 billion neurons is a really significant number of neurons. For reference, elephants have 251 billion neurons, and chimpanzees only have 22 billion neurons. Maybe you have even more than 86 billion neurons. I mean, clearly you're a very smart viewer if you've been watching up to this point. Nerve cells, also known as neurons, serve two primary roles: receiving signals and sending signals. What sorts of signals can a neuron receive? Some neurons detect hormones, signaling molecules which are transported within blood to send signals to other parts of the body. These neurons send signals to other neurons via neurotransmitters in the synaptic cleft between two neurons. This is basically OnlyFans for neurons because the signal only gets sent one way. The first neuron which detects a hormone is called the presynaptic neuron, while the second neuron which detects neurotransmitters is called the postsynaptic neuron. In reality, several neurons could be linked together and be sending neurotransmitters to each other, so the presynaptic neuron may also be a postsynaptic neuron if it's interacting with other neurons, which most neurons do. So how does a nerve receive a signal, and how strong does the signal have to be before the nerve can send its own signal? Most neurons have something called an action potential. This essentially means that enough of its surface receptors must be bound, or transmitting a signal, to trigger the neuron to fire. How do neurons fire? Once the action potential is reached, the neuron will open up its sodium channels and allow sodium ions to rush into the presynaptic neuron. This causes the potential to rise even higher until the sodium channels close and the potassium channels open up, sending potassium ions out of the presynaptic neuron and decreasing the potential of the neuron. When this occurs, vesicles containing neurotransmitters are released from the cytoskeleton of the presynaptic neuron, and the vesicles fuse with the cell membrane and dump their contents into the synaptic cleft. Vesicles are like a tiny little ball which can fuse with the cell membrane to release the contents of the vesicle into the synaptic cleft. Once the neurotransmitters are in the synaptic cleft, the postsynaptic neuron is able to detect these neurotransmitters and send signals on to other neurons in the body. But what happens to all the neurotransmitters in the synaptic cleft? Sometimes, the presynaptic neuron is able to reuptake the neurotransmitters and reuse them. Other times, the neurotransmitters are broken down by enzymes in the synaptic cleft, and the components can be reuptaken and reused by the presynaptic neuron. It's worth noting that different neurons detect different hormones and neurotransmitters, which makes them able to react differently to different chemical signals sent throughout the body. Some examples of hormones which are detected by certain neurons include proteins and peptides, such as oxytocin and insulin, amino acid derivatives, such as melatonin or thyroxine, steroids, such as estradiol or testosterone, eicosanoids, such as arachidonic acid and prostaglandin, as well as gases, such as ethylene and nitric oxide. As we said earlier, you might be detecting a hormone from the body, or you might be receiving a signal from another neuron in the form of a neurotransmitter. So some examples of neurotransmitters which can be detected by neurons include amino acids, such as glutamate and glycine, monoamines, such as serotonin and dopamine, peptides, such as substance P and opioids, purines, such as ATP and GTP, gases, such as nitric oxide and carbon monoxide. This is a perfect moment to tell you about this video's sponsor, HelloFresh. HelloFresh is the best type of HF I've discussed on the channel, and it's honestly the only type of HF I would consume. HelloFresh makes mealtime easy with delicious recipes made with fresh, wholesome ingredients that are delivered right to your door. Did you know that HelloFresh has 40 weekly recipes to choose from? They also have over 100 seasonal and convenience items. Take your pick from meals like soy glazed salmon with rice or mushroom and chive risotto. I'm not gonna lie, salmon and rice? That sounds pretty good. With HelloFresh, you're getting seasonal ingredients picked at peak ripeness for quality you can taste. Their ingredients travel from the farm to your home in less than 7 days, so you know they're fresh. Use my link or go to HelloFresh.com and use code PODCHEMISTMAR60 for 60% off plus free shipping. That way, they'll know you came from here. I want to thank HelloFresh for their support of this channel. Now that we have discussed how neurons work, we can get to the tier list. Neurons are post-mitotic cells, which means that damage to nerve cells can be irreversible, depending on the type of damage that is done. Some temporary disruptions can be treated with an antidote, but not all neurotoxins can be treated. Let's start with the first molecule in our tier list, ethanol. Ethanol is your typical drinking alcohol. Ethanol can be toxic when consumed chronically at high doses. That's because it can cause thiamine deficiency. When the body is deficient of thiamine, that means that it's harder for the mitochondria to metabolize glucose. This ultimately results in the production of more reactive oxygen species, which can do irreversible damage to neurons. Since neurons are post-mitotic, they never get replaced, and oxidative damage to neurons is permanent. There are several different reactive oxygen species that can do damage, such as superoxide, hydrogen peroxide, and hydroxyl radicals, all of which lead to neurotoxicity. Normally, our body has a mechanism to break down reactive oxygen species, but sometimes specific neurotoxic compounds can inhibit that, and that would prevent the breakdown of reactive oxygen species, even ones that would normally be produced during cellular processes, leading to neurotoxicity. So ethanol is going to cause thiamine deficiency. This is kind of an interesting one because it doesn't interfere with any of the nerve functions that we've discussed so far, but it can still cause neurotoxicity indirectly. The ethanol itself isn't causing the neurotoxicity. The ethanol is causing depletion of thiamine. The depletion of thiamine leads to the formation of more reactive oxygen species than usual, and that causes neurotoxicity. So overall, I wouldn't say ethanol is that neurotoxic compared to some of these which have very high acute neurotoxicity. This one's chronic, and it can be managed by not drinking large amounts of alcohol, minimizing your exposure, and ensuring that you have enough thiamine to prevent thiamine deficiency. Ethanol overall, can it cause neurotoxicity? Yes, but I'm going to put it in D tier because compared to some of the other ones on here, it just isn't that neurotoxic. Speaking of that neurotoxic, let's talk about Tetrodotoxin. This is a neurotoxin produced by a specific symbiotic bacteria that are found in specific fish, such as the pufferfish. Tetrodotoxin is a sodium channel blocker, which means that the initial sodium influx into the neuron as it's about to fire isn't able to occur because that channel gets blocked by tetrodotoxin. This effectively means that the nerve can't fire, and so this would cause paralysis. Exposure to pufferfish poison can be lethal. Therapy is supportive based on symptoms, with aggressive early airway management. If ingested, treatment can consist of emptying the stomach, feeding the victim activated charcoal to bind the toxin, and taking standard life support measures to keep the victim alive until the effect of the poison has wore off. So this one's no joke. This is a pretty toxic neurotoxin, and if you don't have a doctor to manage your symptoms, that's it. This is going to go, I think, in A tier because it's super serious if you get poisoned with tetrodotoxin, but there are definitely worse ones that you could be exposed to. Botulinum toxin, also known as Botox. Botox is a peptide, and it's a dimer of a heavy chain and a light chain. The heavy chain of Botox binds to the presynaptic nerve terminal, and the light chain has the active toxin, a zinc metalloprotease. This is a zinc-containing enzyme that's able to cleave the host protein, SNAP25, which is a snare protein, and once this has been cleaved, it prevents the neuron from being able to fuse vesicles with the cell membrane. So this essentially prevents the nerve from sending neurotransmitters to the postsynaptic neuron, since the vesicles can no longer fuse with the cell membrane. Oftentimes, people take Botox to permanently relax muscles in their face. This will result in the appearance of fewer wrinkles. It does have legitimate medical uses, but man, if you get that in the wrong spot, you're gonna have a real bad time. Since the nerve also can't fire the signals, same thing, we get paralysis, which is why we have the relaxation of muscles impacted by Botox. Botox is another really toxic one overall. We can put it into A tier. Next, we have Tetraethylammonium. This is kind of an obscure one if you're not a synthetic chemist, but occasionally we come across tetraethylammonium salts during research. Tetraethylammonium is a neurotoxin because it blocks voltage-dependent potassium channels in nerves. If you recall, after the sodium influx into a neuron, this is followed by a potassium efflux out of the presynaptic neuron. Additionally, tetraethylammonium is a competitive inhibitor at nicotinic acetylcholine receptors. In terms of exposure, it seems like you can recover from treatment relatively easily. As long as this isn't done at insanely high levels, not too much permanent damage is done. I think this one overall is probably less significant than some of the other ones on here. I'm going to put this one into E tier, which is appropriate because it's tetraethyl. Next, methylmercury. Methylmercury is a chemical that frequently bioaccumulates in food chains. Predators, which consume other animals in the food chain, tend to have higher amounts of methylmercury in their body compared to non-predator species such as herbivores. One place that we frequently find this is in fish, especially in fish that eat other fish. We live in a fish-eat-fish world. Once methylmercury is introduced into the body, it's able to cross the blood-brain barrier by binding to cysteine residues in LAT transporters. LAT transporters are a way for our body to bring neurotransmitters through the blood-brain barrier. A cysteine residue has a sulfur in it, and that sulfur really likes mercury. And so the interaction between the mercury and the sulfur, which is due to a soft Lewis base, soft Lewis acid interaction, causes the methylmercury to be transported into the brain. Once this is in the brain, this mercury really likes thiol residues. It really likes sulfur, and so it interferes with sulfur residues in lots of different enzymes, including enzymes involved in the breakdown of reactive oxygen species. Similar to ethanol, via its thiamine deficiency, methylmercury causes more reactive oxygen species to be generated, ultimately leading to more oxidative stress. So this causes more oxidative stress. It's a chronic concern. I think as long as you're not consuming lots of mercury-laden food, it's going to be fairly low risk. It's good to keep in mind not to eat too many foods with high levels of mercury. Oftentimes we have governmental bodies which regulate the amounts of mercury in our foods, so you're probably not needing to measure this yourself. Just don't go crazy eating a ton of top predator fish that might have more mercury than you want to be exposed to. I'm going to put methylmercury into D tier, since it has a similar mechanism to the toxicity of ethanol. I didn't really comment on this, but yes, you can just have like a mercury 1 bound to a carbon, and that's totally fine. Mercury is a really weird element. It likes to do a lot of different chemistry. It's a little bit like palladium in terms of just kind of doing whatever it wants all the time. The next molecule here is this really complex looking bis-indole alkaloid toxiferine. You might think this is just some obscure chemical that no one needs to know about, but in reality, this was historically used as an arrow poison in Central and South America. This is one of the most toxic plant alkaloids, with an LD50 in mice of 10 to 60 micrograms per kilogram by IV. This is a very, very potent alkaloid. This is found in a plant known as bushrope, also known as devil doer. The mechanism of toxiferine's toxicity is as a nicotinic acetylcholine receptor antagonist. While this is really toxic, it can be treated with neostigmine to reverse the paralysis. Since this is a nicotinic acetylcholine receptor antagonist, this blocks the postsynaptic neuron from receiving the acetylcholine signal from the presynaptic neuron. So this is another one that causes paralysis. You can recover from this if poisoned, but recovery can take up to 8 hours. And if poisoning is severe enough, you could definitely die. This one's pretty scary. It's one of the most toxic plant alkaloids that exists. I think this one has to go right into S tier. The next chemical on this list might be a bit of a surprise to you. This is n-Hexane. N-Hexane is a very common solvent, and it's a natural component of petrochemical products, such as gasoline. This one's neurotoxic for a surprising reason. This itself isn't a neurotoxin, but once it's converted into toxic metabolites, it causes neurotoxic effects. N-Hexane can be oxidized at the second carbon on both sides of the chain. Initially, it's oxidized to 2-hexanol, and subsequently to 2,5-hexanediol. 2,5-hexanediol can be readily oxidized to 2,5-hexanedione. This might look rather innocuous to you, but if you're a synthetic chemist, you can imagine what would happen if you mix this with an amine. It would form a perol, and that's exactly what this does in the body as well. It forms perol as it reacts with lysine residues in axonal proteins. So if there's a random nitrogen residue available in the axon, it can react and form a perol. Once these perols form, they can oxidize and cause cross-linking between the perols. This leads to proteins denaturing and disrupts axonal transport and function, causing permanent damage to nerve cells. While this is a concern for n-Hexane, n-heptane doesn't do the same thing because the corresponding 2,6-heptanedione isn't neurotoxic. So n-heptane is a suitable replacement for n-Hexane due to its lack of neurotoxic properties. While we're only talking about neurotoxicity in this tier list, it's important to consider that some of these compounds could still have other toxic effects aside from their neurotoxicity. So disrupting intracellular processes is certainly a concern, but it's an interesting mechanism that's distinct from the other chemicals on this list. Now, in terms of overall neurotoxicity, I think this is rather low unless you're being exposed to a really high amount of hexane in your occupation or your day-to-day life. So n-Hexane, I think it can go a little bit lower. I think it's probably like E tier, which is appropriate because it ends with an E. Next, we have MPP. MPP is actually a herbicide known as cyperquat. MPP was discovered to be a neurotoxin to dopaminergic neurons, as there was a batch of this synthetic opioid drug called MPPP. There was an impurity in MPPP called MTPT, and this is readily oxidized in the body to MPP+. What MPP+ does is it interferes with oxidative phosphorylation in the mitochondria, the US stock exchange of the cell. In this case, we're discussing in the context of neurons. Once the oxidative phosphorylation has been disrupted, this leads to the formation of more reactive oxygen species, as well as by inhibiting the production of ATP, ultimately leading to cell death. So there's a high acute risk, but there's also concerns about chronic exposure. As more reactive oxygen species are generated, this can cause permanent damage to dopaminergic neurons. MTPT is able to be oxidized by an enzyme in the mitochondria called monoamine oxidase B. This is what causes MPP+ to be formed in neurons, and this is why it's generated in neurons and causes irreversible damage. MPP+, this one's a pretty neurotoxic one. I think this one can go right into S tier. In fact, oftentimes this is used to induce Parkinson's-like symptoms. The next toxin on this list is a tropane derivative called Anatoxin-A, also known as very fast death factor. This is a toxin that's produced by various cyanobacteria, and the main route of exposure to people is through the consumption of contaminated water, as Anatoxin-A is rapidly absorbed in the gastrointestinal tract. The mechanism of neurotoxicity for Anatoxin-A is through the agonism of nicotinic acetylcholine receptors in the central nervous system, as well as ones in the neuromuscular junction. The issue is that Anatoxin-A binds irreversibly to the receptor, forcing it to continue signaling, eventually making the receptor become desensitized, which blocks neuromuscular transmission. In the case of Anatoxin-A, acute risk seems to be the main concern, and I wasn't able to find too much information about chronic toxicity concerns. So if you're consuming water that could be affected by Anatoxin-A, it's extremely serious. It's predicted that a lethal dose in humans would be in the range of around 5 milligrams. For that reason, I think Anatoxin-A probably belongs in A tier. The next neurotoxin is actually a recently discovered one. This is called Caramboxin. Caramboxin is found in starfruit. This toxin is excreted by the kidneys. So if someone has kidney issues and they consume starfruit, they could have severe issues or death from consuming even one starfruit. However, it's not just due to the neurotoxicity, it's also due to the nephrotoxicity, which is the toxicity towards the kidneys. Caramboxin isn't nephrotoxic, but starfruit has large amounts of oxalates, and this can cause nephrotoxicity, especially for people who have compromised kidneys. For someone with regular kidneys, consuming 4 to 6 starfruit would be enough to cause nephrotoxicity, but to get neurotoxic effects from Caramboxin, you need to eat around 15 starfruits. Caramboxin is an agonist of both NMDA and AMPA glutamatergic ionotropic receptors with potent excitatory, convulsant, and neurodegenerative properties. This was recently discovered, so an in-depth study of the full mechanism of toxicity hasn't been completed yet. If you need to eat 15 starfruits to get Caramboxin to toxic levels, I don't think you're going to get neurotoxic effects unless you have compromised kidneys. I'd be surprised if I've eaten 15 starfruits in my life, let alone in one session. Caramboxin, it's not too neurotoxic, but it probably needs some more research. We can put it into F tier because eating 15 starfruits is pretty insane. Next, we have Turbocurarine chloride. This is another charged compound that has been historically used as an arrow poison. In the mid-1900s, it was used in conjunction with an anesthetic to provide skeletal muscle relaxation during surgery or mechanical ventilation. Turbocurarine chloride acts as an antagonist for the nicotinic acetylcholine receptor. Since it antagonizes the postsynaptic neuron, the nerve isn't able to send its signal, which leads to paralysis. This one was used as an arrow poison. It's a rather toxic one. I think it can go right into S tier. This compound with all these various different double bonds is called Domoic acid. Domoic acid is produced by algae and accumulates in shellfish, sardines, and anchovies. This is known for its ability to cause amnesic shellfish poisoning. This compound is excitotoxic against ionotropic glutamate receptors at both sides of the synapse. What this means is that it impacts calcium levels in the cell, and this leads to the production of more reactive oxygen species. Because more reactive oxygen species are generated, this will cause more oxidative damage to the neuron, which will be irreversible. There's no antidote known for domoic acid, but medical treatment can lessen the severity of symptoms. Unfortunately, this is a very heat-resistant chemical, so you can't cook it to get rid of it. It'll just persist. So this is a fairly toxic chemical. The generation of reactive oxygen species is undesired, but it's clear that it's worse than ethanol or methylmercury. I think this one can probably go in B tier, because getting exposed to this is no joke. Speaking of no joke, strychnine. Strychnine is a chemical that you might very well be familiar with, and it's probably one of the few alkaloids on this list that most of your family members would also be familiar with. Strychnine is most commonly found in the strychnine tree. This plant also contains a similar molecule called brucine, which is almost identical, it's just got a couple extra methoxy groups. This is still occasionally used as a pesticide, and there's even people crazy enough to use it as a performance enhancer, as it's been used historically in small doses to strengthen muscle contractions. The mechanism of strychnine's neurotoxicity is as an antagonist of the glycine receptor, which is a ligand-gated chloride channel. Strychnine is an antagonist of glycine, and it binds non-covalently to the same receptor, preventing the inhibitory effects of glycine on the postsynaptic neuron. Therefore, action potentials are triggered with lower levels of excitatory neurotransmitters. When the inhibitory signals are prevented, the motor neurons are more easily activated, and the victim has spastic muscle contractions, resulting in death by asphyxiation. There is no treatment, but recovery from exposure is possible with early supportive medical treatment. So basically, your body has to get rid of this, and we just need to keep you alive till you survive. So spastic muscle contractions, in this context, essentially means that the nerves fire like bananas. That's not great. This can go right into S tier, which is appropriate because it starts with an S. Here we have one of the longest molecules we've ever talked about in a tier list. This is Annonacin. Annonacin is a chemical compound found in some fruits, such as the pawpaw, custard apples, and soursop. Annonacin is a disabling and potentially lethal neurotoxin. This compound interacts with the NADH dehydrogenase enzyme present in mitochondria. Interfering with this enzyme disables the cell's ability to produce ATP through oxidative phosphorylation. This ultimately leads to the death of the cell or necrosis. This compound is 100 times more toxic than MPP+, but it has the same mechanism. So MPP+ is S tier. Annonacin also belongs in S tier. It's 100 times more toxic, and its structure is super innocuous. This just looks like a long chain, guys, with a couple OH groups. This looks like super chill, but it is like not chill at all. It's like the least chill. You might think that this is a pretty depressing video, but hey, hopefully you're starting to understand how nerves work. If this video is making you nervous, I must be doing something right. Tetraethylead is an additive that's been used historically as an anti-knocking agent in leaded gas. This is still used in some specialized applications, but leaded gasoline has more or less been outlawed for most consumer use. When introduced into the body, tetraethylead is metabolized by the liver to form triethylead, which is considered to be the primary toxic metabolite. Triethylead is able to cross the blood-brain barrier, presumably through similar lat transport, but I wasn't able to find as much research into the neurotoxicity of tetraethylead. The half-life of this compound in the brain is 500 days, meaning that once it goes in there, it's going to be there for a nice long while. Trialkyl lead compounds are believed to disturb the chloride ion flux and transport of the neurons. This type of ionic change would tend to reverse the GABAergic neuron's hyperpolarization action, which allows neurons to be depolarized with exaggerated excitation, which would in turn induce neuronal damage. So a 500-day half-life in the brain is pretty awful, meaning that it's going to keep disrupting processes for a very long time. When GABA is hyperpolarized, it means that more calcium is going to be let into the cell. More calcium into the cell means that more reactive oxygen species, meaning that there's more oxidative damage that's permanent. Tetraethylead, you can also go right into S tier. Here we have Chlorpyrifos. We made a whole video on nerve agents previously on the channel, and in that video, I misspoke. I said that nerve agents are acetylcholine antagonists. Nerve agents are these things that inhibit this molecule called acetylcholine, but actually, nerve agents are acetylcholinesterase inhibitors, meaning that the breakdown of acetylcholine is prevented. Chlorpyrifos is a similar nerve agent that's used on crops, animals, and buildings. This is also an acetylcholinesterase inhibitor, and it's considered moderately hazardous to humans by the World Health Organization as a Class 2 agent based on acute toxicity information dating all the way back to 1999. Exposure surpassing recommended levels has been linked to neurological effects, persistent developmental disorders, and autoimmune disorders. Additionally, it seems like there's concern about exposure during the mental development of children. This is especially neurotoxic during development. It irreversibly inhibits acetylcholinesterase, but it seems to also have some other mechanisms of toxicity. This is another extremely toxic one. This can go right into S tier. Now, when acetylcholinesterase is inhibited, oftentimes this can be treated with certain antidotes, but since several of these compounds target lots of cholinesterases, not just acetylcholinesterase, there can be several different mechanisms of toxicity, and it can be challenging to combat all of them. And it's also challenging to say, as a rule of thumb, you can always use this antidote for all of these nerve agents. Oftentimes, treatment needs to be tried by a trained medical professional who has far more experience than I do. Next, we have this strange bicyclic compound called TBPO. This is a chemical that's only used in research, and I mostly just included this one for researchers. This is a GABA receptor antagonist, and unlike most phosphorus esters, this doesn't inhibit acetylcholinesterase, although it is still neurotoxic. This is the most toxic bicyclic phosphate known, with an LD50 of 36 micrograms per kilogram in mice. There are several other compounds with similar structures, which are just as toxic. You don't need to have a phosphorus here. Even if there was a carbon here and we had an orthoester, such as some of the ones I'm going to show on screen now, you can still have similar neurotoxic effects. There are other ones where a nitrogen's present. Sometimes there's also a silane present, which is able to coordinate to that nitrogen, and this sort of bicyclic structure with these oxygens is a red flag for potential neurotoxicity. This is a pretty scary one. This one should also go right into S tier. S tier is looking pretty full, but this is a neurotoxin tier list after all. If you don't think that these belong in S tier, maybe if you got hit by a poison arrowhead, you might change your mind. If you would willingly be exposed to anything in S tier, I think you're probably insane. Next, we have Neosaxitoxin, and I have to say, this molecule sure is Neosaxy. This chemical is one of the paralytic shellfish. I can't say shellfish for some reason. I'm so selfish. This molecule is so selfish because it's one of the paralytic shellfish toxins. Another member of this class is Saxitoxin. Neosaxitoxin and other paralytic shellfish toxins are produced by several species of marine dinoflagellates and freshwater cyanobacteria, as well as blue-green algae. It's been associated with toxic algal blooms that can have severe impacts on marine ecosystems. The mechanism of toxicity for Neosaxitoxin is through its ability to reversibly bind to certain voltage-gated sodium channels. These are the channels that allow the initial sodium influx into the cell. By inhibiting these, it effectively paralyzes you. The solution to this is to keep the person alive until it wears off. Additionally, there's been some people looking at using this as a potential new local anesthetic, since it can wear off. So Neosaxitoxin, this is another one that can paralyze you. Since this one has potential as a new local anesthetic, I think this is probably a little bit better than some of the other poisons on here. Why don't we put Neosaxitoxin into A tier? I'm just going to gloss over how completely cursed Neosaxitoxin looks. If you show this to an organic chemist and ask them to make it, they're going to quit on day one. The next molecule is carbon disulfide. Carbon disulfide is a common solvent and is frequently used in mineral flotation. Carbon disulfide reacts with amines in the body and forms dithiocarbamates, which deplete zinc and copper from neurons. These also tend to react with pyridoxine, which is an important cofactor. The depletion of these metals, zinc and copper, leads to the inactivation of several key enzymes. The neurotoxicity seems to occur as a result of increased oxidative stress. Since the oxidative stress is increased, this results in permanent damage to neurons. Compared to some of the other ones on here, this damage should be more reversible, since exposure to high amounts of carbon disulfide is relatively rare unless you have occupational hazards. I'm going to assume that this is roughly about as bad as hexane, so we can put it into E tier. It probably goes without saying that any of the stuff on this list is still probably concerning, and you really shouldn't be exposed to them. S tier is looking like it could use a bit more room, so we're going to use the remainder of the space on the top here as an extension of S tier, because a couple of the ones here definitely belong in S tier. TETS has historically been used as a rodenticide, except it had a problem. It killed rodent populations for up to 4 years. TETS binds to neuronal GABA-gated chloride channels, often causing lethal seizures. No antidote is known, and it's been banned since 1984. This chemical is so simple that it's scary. This has been historically used in 2 mass poisonings in China, in April 2004 and another in September 2002. This has no known antidote, and it's extremely potent. This definitely belongs right in S tier. The next molecule is Aldicarb. This is another nerve agent. Aldicarb is one of the most widely used pesticides internationally, and it's also one of the most environmentally toxic. This is a carbamate insecticide, which is this group on the left here, and it's typically used as a nematicide to kill nematodes.
Oh, dang nematodes!
Aldicarb is a fast-acting cholinesterase inhibitor. It inhibits cholinesterases, which is a broad term used to refer to several different cholinesterases, leading to the accumulation of cholinesters, like acetylcholine, in synapses. Exposure to high amounts of Aldicarb can cause weakness, blurred vision, headache, nausea, tearing, sweating, and tremors in humans. High doses can be fatal because it can paralyze the respiratory system. Now, I don't know in terms of its chronic toxicity. I think since this is so widely used, that's probably something someone should be looking into if they're not already. It may belong in S tier, but because I don't know quite enough about this, I'm going to put it in A tier for good measure. If you know anything about Aldicarb's toxicity, or if you have any stories about people who've worked with it, I'd love to hear those stories down in the comments. The next molecule is Batrachotoxin. Batrachotoxin is known as the dart frog poison, but it's actually found in 4 different beetle species, all from the genus Coracine. Poison dart frogs have been used to make poison darts historically. This is because the alkaloid tends to concentrate in their skin. Batrachotoxin's toxicity is due to its ability to bind irreversibly to sodium channels. Batrachotoxin binds to and irreversibly opens sodium channels of nerve cells, preventing them from closing, resulting in paralysis and death. No antidote is known, but the effects of it can be reversed with Tetrodotoxin, which has the opposite effect. So once the sodium starts going into the cell, it just keeps going and keeps going. That's pretty awful. Poison dart frog, this has been used to poison darts. I think we can throw this one right into S tier. The next spicy molecule here is Resiniferatoxin. Resiniferatoxin is one of the spiciest chemicals ever to be discovered. It has a score of 16 billion Scoville heat units, making pure Resiniferatoxin about 500 to 1,000 times hotter than pure capsaicin. This is naturally found in the resin spurge. This is a spicy molecule because it targets TRPV1. TRPV1 is involved with inflammation and pain. It's an ion channel in the plasma membrane of sensory neurons, and stimulation of this ion channel by resiniferatoxin causes the ion channel to become permeable to cations, especially calcium. The influx of cations causes the neuron to depolarize, transmitting signals similar to those that would be transmitted if the tissue was burned or damaged. This stimulation is followed by desensitization and analgesia, in part because the nerve endings die from calcium overload. So exposure to this level of spiciness can even cause neurotoxicity, which is pretty wild. I think this one can go right into A tier, because ooh, that's a spicy boy. For some reason, I forgot to add one of these chemicals onto the tier list, so we'll just add it on now. 5,7-dihydroxytryptamine is a neurotoxin that's used in research to destroy serotonergic neurons in the brain. This is what serotonin looks like, and you can see the structural similarity to dihydroxytryptamine. This one's mostly just of interest to research, but it's definitely neurotoxic to serotonergic neurons. Since this does destroy neurons, for sure, and it's specific, this one's certainly very neurotoxic. We can put this into S tier. Similar to dihydroxytryptamine, we have oxidopamine, another hydroxylated neurotransmitter derivative. This is used to destroy dopaminergic and noradrenergic neurons in the brain. Those are neurons for dopamine as well as noradrenaline. In research, it's used to induce Parkinsonism, which is Parkinson's-like diseases, in laboratory animals. This one destroys dopaminergic neurons, specifically used to cause Parkinson's in animals. This can also go right into S tier. Another neurotransmitter-like chemical is para-chloroamphetamine. This is a designer drug which is neurotoxic, and it's thought to be neurotoxic due to the unrestrained release of both serotonin and dopamine by one of its metabolites. This has also been used as a neurotoxin by neurobiologists to selectively kill serotonergic neurons for research purposes, similar to how 6-hydroxydopamine is used to kill dopaminergic neurons. This one's pretty neurotoxic. I don't know if it's quite as severe in humans as it is in research, but I'm going to go out on a limb and say this one's probably around B or C tier, since one of the metabolites is the actual cause of neurotoxicity. Last but not least, we have Penitrem A. This is a very wild structure. Look at all these rings here. This would be a nightmare to synthesize. Penitrem A is a mycotoxin produced by certain species of Aspergillus, claviceps, and Penicillium, which can be found growing on various plant species such as ryegrass. Penitrem A does the same thing that some of the other ones do. It impairs GABAergic amino acid neurotransmission and antagonizes high conductance calcium 2 activated potassium channels in both humans and animals. Penitrem A also induces the production of reactive oxygen species, leading to irreversible damage, as with some of the other ones here. This is a potential concern as it could be a mycotoxin present in our food, and it leads to the generation of more reactive oxygen species. For that reason, I'm going to put it into A tier, as this is severely toxic. So you can see most of the neurotoxins that we've discussed in this tier list are rated very high. That's because most neurotoxins do a lot of damage right away, or they put you at acute risk of death, something you generally want to avoid. So hopefully this video has made it easier for you to understand how neurons work. There are many other neurotoxins aside from the ones we discussed in this video, as there's many ways to disrupt neuronal function. I'd like to give a huge shout out to David, also known as Reform Labs, as well as Nicholas, also known as Aldos, who helped in the production of this video. I hope you've enjoyed this tier list. Thanks for watching, and I hope you have a great day.